So you heard about transfer membrane protein prediction evaluation last time, I believe. This is one of the slides that Burkhard showed. He gave some kind of outlook that there may be a way for improving the current level of performance, and this blue bar here representing TMSEG is exactly what the method that I'm going to talk about today. So yeah, we just submitted it to Proteins. It's not published yet, but hopefully soon it will be out. So you have already seen Tuesday then probably, that there's already quite a few transfer membrane helix predictors, and in that evaluation we looked at only 12. In reality there's more than 50 easily. So you may ask, why do we want yet another new method? Aren't there enough already? And so one of the reasons is that, of course, data for transmembrane proteins is very scarce. As you know, determining their 3D structure is extremely hard, much harder compared to soluble proteins. So we have very little data, and any additional data that we can get, we expect to improve our prediction performance. So we want to build predictors based on better data, on more data. You could technically just use a method that you have developed previously and give it a new data set and retrain the method on this new data set. However, no one seems to be doing that for some reason. I'm not sure actually, but it's really uncommon to do, although it's a completely valid approach. And for our case in particular, so we do have a prediction method from this lab, from Burkhard himself actually, which he wrote decades ago, but that is so old that for us it actually did make sense to write a new method, and that is TMSEG. Another point is that I don't believe you discussed it in that much detail, but many of the methods currently out there are relatively sophisticated in the amount of machine learning that they apply. They have a very high number of free parameters, and given how little data we actually have, that could easily lead to overfitting and overtraining the data. So if we can do it in a more simple way, we would very much prefer that to having something that is extremely highly optimized on a data set for which we don't even know if it even represents all of the reality, and which actually is very small. And then finally, that also goes in with the third point. So the more complicated you make your whole machine learning, the higher the runtime usually gets in addition. And that, of course, if you only have 10 proteins of interest that you want to predict, then it's fine that MEMSET, for example, predicts, I believe, a third of a sequence in a minute or so. If you only have 10, that's good. But if you want to predict the whole proteome, you want something that's a bit faster, ideally. So we try to improve on that as well. Okay, so first of all, we need to collect a new data set. The protocol for this data set collection is pretty much exactly what we also did for the evaluation that you already heard about, only that this was done a bit later, so the data set is a bit larger, but the idea is the same. We need 3D structures. From 3D structures, you can infer kind of where the membrane is, and through that also where the membrane helices are. There's two big databases that do this, OPM and PDBTM. And since you do not explicitly see the membrane in the structure, you do not know exactly which residues are in part of the membrane helix or not. So the assignments by these two databases differ. And we can't really say that one is more true than the other, because we simply don't know. And actually they may both be right in some cases when the protein actually does move a bit in the membrane. That's how it is in reality. So what we do, we use the assignments from both of them, and both of them are used for training. Same as for the evaluation, we assume that both are correct. So we use OPM as well as PDBTM annotations. Then the next point that also is the same as an evaluation, the sequence that you can infer from the atom coordinates of the PDB file is not necessarily the natural sequence. So for determining the structure, there may have been mutagenesis performed to make it crystallize easier. It may only be a fragment of the reality. They may have cut out parts. They may have added some parts. So it's easier to perform the structure. In general, this is not necessarily what you would consider the normal state of the protein in terms of sequence. So what we do is we map the atom coordinate sequence to the seq-res sequence that is denoted in the PDB file. And then from that we go on to map to the sequence that we find in UniProt, which to us is the more realistic application. So you always have to think of what would be the type of sequence that you actually put into your method that you want to predict later on. And you're not going to put in the sequence that is in a PDB structure, because then you don't need to predict anything anymore. So what you have is what we believe the protein looks like, what is inferred from the gene, and that is what's in UniProt. Mapping that is actually not very nice to do if you would were to do it yourself, but luckily there's this SIFT service from the EBI, and that does it relatively straightforward for you. Yeah, and once we have that, we of course have to perform a redundancy reduction on the sequence. For that, we're using UniProt, which uses the HSSP value. You've all seen the curve, so that you should know. Okay. And finally, what we did not do in the evaluation, so we only looked at where are the helices in the protein, but you can also look at the topology of the membrane protein. So the non-helical, the soluble loops, on which side of the membrane are they at? So for that we also need an annotation. By now PDBTM does have that annotation directly in the structure. When we collected this data set, that was not the case yet. So instead we took the annotation from OPM. OPM on the website tells you where the N-terminus is. And then if you download the structure, they added these dummy atoms here, which just represent where they believe the ends of the membrane are. And then you start from the N-terminus and you just look at the 3D coordinates where the residue is in space, and from that infer on which side of the membrane you currently are for every residue. Hey. So that's the one major change to the protocol of the evaluation paper that you've already had before. We in addition also have information about topology now. Okay, and then we need an additional data set. So the first data set tells us where are the helices, but for such a method is not only being applied to transmembrane proteins. So again, if we, for example, apply it to a proteome, we don't know which part is the transmembrane protein and which isn't. So that is also something that the method has to find in the first place. And for that we have an additional bigger data set, which is based on the training set of Signal P. Signal P, I believe you've heard about before, predicts signal peptides, and they have the same issue. So you know, signal peptides have a hydrophobic region, and if you do not account for the fact that signal peptides and transmembrane helices, because of that hydrophobic region, kind of look like the same to machine learning, you get many false positives. And that goes in both directions. So you know about Phobius, for example, which is a transmembrane helix predictor that accounts for signal peptides, so they don't get false positive helix predictions when it's just a signal peptide. But Signal P predicts signal peptides, and it works the other way around as well. So they have to make sure that they don't consider a transmembrane helix as a signal peptide. So they have a data set of proteins which contains soluble proteins that have signal peptides, transmembrane proteins, and transmembrane proteins that have signal peptides. For those, we do not have the 3D structure, so we cannot use this for what we're using the first set for. But we can use it to just determine if the whole sequence is correctly predicted as one of the protein classes. So we collect that set, and we redundancy reduce it first of all against the set that we already have, the 166 transmembrane proteins that have a structure. And then from the resulting part, we also again perform a redundancy reduction on that set to make sure that we have no biases left in the whole training set. And that gives us in total 1,441 proteins. 1,142 of those are soluble. The rest are membrane proteins, and then a subset of those in every case also contains a signal peptide. Right, and then of course to train our method we split the whole thing up. In this case 4 subsets. The cross-validation is performed on 3 subsets. Of course, while splitting up we try to keep the distribution of the input sequences more or less similar between the subsets. So he tried to keep the sequence lengths more or less the same to make sure that there's more or less the same amount of helices in every subset and so on, to minimize biases. And then among the 4 sets, 3 are used for training, and the 4th one he calls a blind set that he took out and is only going to use at the very end for evaluation. So this is all used for training, and we of course do get cross-validation performance from the training, but in addition to that we want to have performance on a completely independent set, and this is what the blind set is going to give us at the end. Right, so TMSEG is using random forests, and because this has never been covered before, just very quickly, what are random forests? Random forests consist of trees, so first of all we have to talk about trees. Classification trees you have probably seen before, so very basically you just have a set of training samples, you have a set of features, and you want to partition this feature space, recursively partition the feature space in a way that you best predict your labels, your class labels that are then found in the leaf nodes of the tree. And how exactly to do that? There's many different algorithms, so you can prune trees, you can cut parts off, you can have different heuristics for determining how you actually split, which feature to choose to split, and so on, but we're not going to talk about any of this, so just the basic idea is this is such a classification tree. It has 3 classes, so the class labels are in the leaf nodes, they're colored here, and then we have 2 features called x1 and x2. And the tree now tries to classify based on these 2 features which of our input samples belongs to which class. And the algorithm, whichever algorithm that was in this case, chose at the root node, it can choose either use x2 or use x1 to distribute the class labels as best as possible to the left and right side of the tree. And in this case it chose x2. And then also it has to choose a threshold value at which to say, okay, anything that is above the threshold goes on this side and anything below goes on the other side. So in this case, x2 being smaller or equal to 0.7 is going to put the label into the left side and everything else onto the right side. And the same thing is visualized here again. So this is our feature space, we just have 2 dimensions, that's why it's easy to see in this case. x2 is on the y-axis, and at 0.7 we have our threshold. So just looking at this visually, you can see that why this probably happens. So most of the 1 classes are now above. That already gives us some kind of distinction. And now we just continue. So if, for example, we look at all of those that are bigger than 0.7, that would be this part up here. And that would be the right side of the tree. Then we have our other feature, x1, x1. And again, we have to now check, okay, so the feature is clear, we only have 1 left, but we have to still find the best threshold to distinguish the 2 classes. And in this case, minus 0.61, which is then represented by this line. And you can see that this quite well actually distinguishes between the class 1 and the class 2. And it's the same thing on the other side, which would be the part down here, where it's not as easy, but there it also kind of tried to put all of the 2s on the left side and class 3 on the right side. And since we start at some point and then work our way down, so this is what this recursive part is, this isn't necessarily an optimal solution. For 2 dimensions we could calculate the optimal solution by some metric that we define, but usually you have much more features, the feature space becomes really large, and then an optimal solution is not really feasible. So that's why in building these trees you recursively partition the feature space and yeah, take that as an approximation. Okay, so we know what trees are. Many trees are forests, which is why the ensemble method that uses trees as its base weak learner are called random forests. The idea is you do not build 1 tree, but many. And for building every tree, you have your set of features, but you don't use the whole feature set. You use a smaller number of features, which you randomly choose for every tree. And then in building the tree, you subsample all of your training data. So you usually also don't use the whole training set, but a part of it. And after that, it's more or less, we're not going to go into details again, but more or less the same. Given that subset of features and training data, you build a tree. And then you just do this many, many times, and then you have many trees, and the ensemble of all of these trees, the prediction from all of the trees is what then gives you your final prediction. So yeah, that's the basic idea. Why do we care about these? So first of all, trees are really fast. I mean, you just look at the feature and then you have a threshold and you go right or left, so that doesn't require a whole amount of calculation. So they can be very optimized, and if you have many trees, of course it becomes slower, but it's still relatively fast compared to a neural network that has hundreds or even thousands of hidden nodes, for example, where there's much more math involved. It's also not a black box, so these trees, you can actually look at the tree. It's not like some hidden nodes where you don't know what they represent and then they are somehow connected. You can actually look. Here you split on this feature because the tree believes this feature is important here. Sure, if you have hundreds of features, it's still going to be hard to kind of keep the overview, but you can technically look at every tree in the forest, which makes them very intuitive in a way. And also, just generally, it happens to be a pretty good model that usually achieves relatively high performance, certainly in our case. So that's why we then applied them. Yeah, and just to give some background again, so they were introduced relatively recently, 2007, I believe, around that time. And since then have gained in popularity. So what they did here is just they mined PubMed, I believe, and looked at all of the machine learning models that appear in papers. And as you can see, random forests are gaining in popularity, so are SVMs, although they began a bit earlier. And neural networks are actually going down. Although this ends in 2010, and with the recent advances of deep learning becoming popular and all, probably they're going to increase again as well. Yeah, and so these are the trees, by the way, that they kind of go up with the forests. Makes sense. Probably in every forest paper there's trees mentioned as well. Okay, so this is the method now that we are developing. It's built, it has 4 steps. And in the first step we use a random forest classifier. It has 100 trees in the forest, so not that much actually. And we always use 9 random features out of our whole feature space to build the single trees. We use a sliding window of 19 residues, which for transformer helix prediction is more or less what you use usually, somewhere around there, also for neural networks, for example. And the output of the tree are 3 different scores. The probability of every residue to either be a signal peptide residue, a transformer helix residue, or a soluble residue. And then simply for calculation reasons, we do not use the probability from 0 to 1, but integers from 0 to 1000, because calculating integers is faster. Sorry. And we don't need the additional accuracy of the floating point. So the 4 digits there are more than enough. And then the result looks something like that. Yeah?
Did I get it right that it's 9 out of 19 residues for the features?
No, sorry. So we have a certain amount of features which we will get to in a few slides. And the features are calculated over a window of 19. Okay. So the average charge over a window of 19, for example, is then 1 feature. Mm-hmm. And we will see how many features we have. Okay. So that's what the result then looks like. On the x-axis we have our sequence, and for every residue we get the scores. Yeah, and as you can see here, for example, and at the start apparently there's probably a signal peptide. We have relatively high scores for signal peptides. And then the rest, there's alternation between high scores for soluble residues or transmembrane helix residues. So we can kind of already see that here is probably helix, then there's some soluble part, then there's a helix again. But this is relatively rough still. Before we get to the next step about the features that you just mentioned, so there's a few global features, the global amino acid composition, which is pretty much always a good idea to take into account. Also the protein length in some bins, not the exact length, because we don't really expect that a protein that's 300 residues long is different from one that's 302 residues long. I mean, if we use the exact length, we would be implying that we believe this is an important difference, and we don't at this point. And then many local features which are calculated over the sliding window. For example, the scores from the PSSM, which we get by blasting against a big database, the distance to the N and C terminus, and several other features related to hydrophobicity, of course, which you know is really important for transformer helices. and also charge, which is important for determining on which side of the membrane we are on. And the same thing more or less goes for polarity, only that those we calculate only over a sliding window of 9 residues, because it just, in the development, it was found out that 19 kind of is, the signal does not really exist anymore on a window of 19. So for these, using 9 proved to be better. That's just, yeah, question.
How do you map like global amino acid composition to that one-dimensional value?
To what, sorry?
To one-dimensional value, because you need to kind of find a cutoff, right, for the tree.
I'm not sure I got it. To how I map it to the window. Because this is not on a window.
I mean, like, how do you represent the global amino acid composition, for example?
In 20 features. So just for every amino acid, how often does it appear?
Oh, right.
So that's what we mean by global amino acid composition.
Oh, right.
And we will, so the actual number of features that all of these are, we will also get to in a second. But before, there's one more additional step that we do. So we have the PSSM score that kind of does take into account conservation for us, but Michael, developing the method, wanted to make it a bit more explicit even for the other features as well. So what he's doing is, this is a PSSM, our sequence is here at the left, and for this calculation we're going to assume that this is the whole sequence. Usually it's done over the whole PSSM, but I can't display this here. So say our sequence was only 5 residues long, then this is our PSSM, and what he does is he looks at in the matrix and counts how many scores, how many substitutions are not predicted, are expected to be conserved from the evolutionary profile, meaning they have a score bigger than 0, and which residues have, which substitutions have a score smaller than 0. And we just count all of these. The zeros are actually ignored because the expectation for the zeros is that they represent the background. So they are neither nor, they don't really tell us anything, so those we are going to ignore. Now if we have that, we take a feature, for example, the amino acid composition for methionine, and we now calculate it, but only, so we calculate it for all conserved substitutions. So what we do is, we look at the column for the methionine and we check how often is there a score bigger than 0, and in this case that's just this one here, so among the 5 that we have, that's just one, and in total we have 16 substitutions that were conserved, so that score would be 1 out of 16. And the same thing we do for the non-conserved positions. In that case it would be 3 out of the whole 79. Again, the 0 we just ignore, both in calculating the total as well as when calculating only the feature that we're going to look at now. Yeah, and just as another example, same thing here for the positive charge, for example. We positive charge in this case is arginine and lysine for us. That really have a strong positive charge, so we look at those 2 columns, and again we check how often is there a score bigger than 0. For arginine there's 1 here, and for lysine there's 1 down here, so that's 2, and all of the others are 8 in total, and that way we essentially use this feature percent positive charge not once but twice, split up by whether the substitutions are conserved or not. So that then gives us our total number of features here at the right. Amino acid composition, I said 20, but now that we split it up, it's 2 times 20. And the same thing for all of these down here. We take all of them just not once, but twice. And these are not affected by this. So this is going to appear again. All that are marked by this PSSMs bigger or smaller than 0, we essentially take twice based on the substitution matrix. Okay, so that's the first step. We have predictions for transmembrane helices. Maybe go back here. So this is where we currently are at. Now, in the next step, we are going to smooth this whole, so the curves were pretty, they jumped around a lot, and we want to smoothen the whole thing a bit. And for that, Michael used a median filter that has only a relatively small window of 5. And we will look at an example of that in a bit. And what he also did is he reduced the scores a bit for overrepresentative classes of soluble residues and transmembrane helix residues. Again, these values are just empirically chosen, so there's no machine learning involved here. And then once we have all of that, every residue, we still have 3 scores, and we choose then for that residue the prediction that has the highest score. So the label of the highest score wins. And then finally, some senseless predictions are removed. So signal peptides that are smaller than 4 residues do not make any sense, which the machine learner did not intrinsically learn. So we just delete them. Delete as in we map it to soluble. And the same thing for transmembrane helices, which are smaller than 7, which is also impossible. So no membrane is that thin that 7 would be enough. So same thing here, we just map them to soluble. Right, so this again visualized. First we have the median filter. So if we look at the colored residues, we have a window size of 5. So this here is now our center residue. We look at the 2 residues to the left and to the right. And we take the median of that window of 5, which in this case is 600. And then afterwards, 600 gets assigned as a score to this residue. And we do this just sliding the window over every score, over every position, and then we get this. And then based on that, on those scores, we do the adjustment I mentioned. So signal peptides are not adjusted. They are the smallest class we have, so they are not touched. But for soluble and for transmembrane helix, we reduce the score a bit, which then gives us these scores. And then for every column, we would check what is the highest score here. And in this case, since in this case, signal peptide is always the highest, so this is at the start of the sequence apparently. And then the final class labels predicted would be signal peptide for all of those. And that's the same thing visualized again. So now we actually have the labels assigned to every residue. Okay. In the third step, the helix predictions are refined by using a neural network this time. It has a relatively small, only 25 hidden nodes. And the main difference here is now that we don't have sequence, so we still do have sequence input, but we don't use a sliding window over the whole sequence or anything like that. Because we already assigned here our segments, so we know where the helices, in this case, where the helix segments are. So all we're going to use as an input now is this segment of the helix, and that means that we can't have any position-specific features anymore. So now we only have features that we can calculate on some segment of a helix that can have variable length. And what we use here is again the amino acid composition of only that small segment, the hydrophobicity, the charged, the percent of the charged residues, and then also here actually the exact length of the segment. So now we're talking about maybe lengths from 10 to 40 or so, and here it actually does make a difference if it's 1 or 2 longer or shorter. So there's no binning here anymore. We actually use the exact length of the segment as an input. All of this goes into a neural network. What we also then do is looking at very long helices. So a helix that's longer than 35 residues is being checked if splitting it up into 2 that have 17 residues would improve the score. And since you are so close to me, I can actually ask you, why do we do this? So for the computer scientists, they're all like hundreds of meters away. But I mean, so why would we do that? First of all, why would we split long helices? Or why would we not split them? I mean, you can argue for both points. That's why we look at both cases.
In some cases, it would make more sense if you split it and it is a multipass membrane protein.
Yeah, so
for other cases, it would be not like that. So.
Yeah, so I mean, one case that you could have is that you have a helix and then maybe just 2 or 3 residues, just really exactly one loop as short as possible, and then immediately helix again. So you could argue that the the small soluble loop signal is so small that the predictor may not capture it, or not without us looking at it specifically again. And that would be a case where we want to split a long helix into 2 helices. But we don't just always do it. So we look at those, but we only use the 2 small helices if the score of the 2 helices is better than the score of the long helix. And that is why, that's because long helices are possible. So not every helix is perpendicular to the membrane. If it's extremely tilted in the membrane, then 35 residues is possible. So if the score is still as higher, if there really is a high hydrophobicity signal all throughout the sequence, then we leave it long. So that doesn't appear that often, but there are proteins that look like this. And finally, the endpoints are adjusted. So that, in a way, also is related to the fact that, again, we have annotations from OPM and PDBTM, so we don't really know where exactly the membrane ends, and there's some variation. And this simply tries out 3 residues at both ends in both directions, shorter and longer, what seems to give us the best score, just to improve the prediction, to some degree. To how much we will actually see later. So that's the result after the 3rd step. In this case, the helix was split up, so we had this really long helix signal here, which is why we assigned a very long helix over the class labels, but apparently here the score actually got better by splitting it up. And you can, well, maybe see that there's this dip here, so maybe here there's just a soluble loop, a really short one. And that's why it was split up into 2 helices. Although that was not a good example, because so this is actually not what this predictor sees. So it only gets the input what we calculate from the segment. So we kind of see something here which is then apparently also captured in the features we have from the segment. That's how I could say it. And then as you can see also the endpoints were adjusted a bit. So this helix ended here, but now this one ends here, and the same here, so they are shifted a bit further out. Okay, the 4th step, we now have our finished helix prediction, but we still need to know the topology, so the soluble loops, which side of the membrane are they on. For that we use a random forest again. And what we can do, so we don't know which side is outside, which side is inside, but we can call them side 1 and 2. So if this is side 1, then that's 2, then that's 1, then that's 2. So we assign the 2 sides. Assuming that our helices are correct, we have to assume this at this point. I mean, we're not going to get any additional information. We did our best, and doing that then we can calculate these features, the amino acid composition, the % positive charge, and the difference between the positive charge between the sides. And we can look at this conserved, non-conserved, but also side 1 and side 2, so that's why we have 2 times 2 for these features up here. Not for the one down here, because that's the difference between the 2 sides, so that you can only do once for the 2 sides. And we look at the difference between the difference of the positive charge between the 2 sides, of course, because of the idea of the positive inside rule, which you've heard about, that the inside loops usually have more positive charges than the outside loops. We don't explicitly model that rule, but we expect that the machine learner captures this if we give it the feature. So, there's more. Oh yeah, one more thing. We do not look, when we calculate these features for the soluble parts, for the non-transmembrane loops, we do not look at the whole loop. So there are transmembrane proteins where such a loop can be hundreds of residues, which has its own function and goes on completely elsewhere. In that case, we do not want to integrate this here, because that would kind of weaken the signal. I mean, this is its own domain then in some right, and that all we want to see here is which side of the membrane are we on. And for that, it actually is enough if we only look at the border cases. So actually, if you add residues into the membrane itself, because they can shift a bit, and the first few residues in the membrane are also important, and then the 15 residues outside of the helix, but nothing else. So whatever then is also attached outside, we don't really care about. That's not important for determining which side of the membrane we are on. So only for those we calculate the features we saw before. And then we predict where do we believe the N-terminus is. So that's the output of the random forest. And once we have that, Again, assuming our helices are correct, we can just assign every side, because then every helix is assumed to switch the topology, and yeah, so from there on it's trivial to assign the sides. Only if we predict a signal peptide, they are usually, so the residues following the signal peptide are usually outside. So if a signal peptide is predicted, then this N-terminus prediction thing is actually not performed. Yeah. So that's then the final thing. Here we have a signal peptide. So these residues are outside of the membrane. And then it just switches over every time. And again, here he also highlighted the positive charges. Yeah, so they would be used as an input feature to the random forest. But it's not part of the output or anything. Yeah, it's a bit confusing to have that there. Okay, so that's the final predictor. Now we want to know how it performs. You already know per residue scores are not very good, because you can have issues where you have, for example, a transforming helix predicted, but then in the middle of the helix there's 1 or 2 residues which are predicted soluble. If you predict the accuracy per residue, for example, if you predict how many helix residues do I have correct, how many soluble residues do I have correct, then over the whole protein these 2 residues are not going to make a big difference. But biologically, of course, it makes no sense to have them in there. So we want to kind of capture cases like that. Which is why we don't look at single residues, we look at, are the segments correct? Are the helices correctly placed in one piece, so to say? And then what we actually want to know is that true for the whole protein? Because just missing one helix, if we assume that we can just switch topology, missing one helix is a very crucial mistake, because everything afterwards is then going to be wrong. And knowing which loop is on which side is extremely important for the function of the protein. So what we actually would want is that I mean, it's a very strict score, but what we actually want is that we get every helix in the protein correct. And that is what QOK represents, and that's also what you saw last time. Yeah, so that's QOK again. We calculate the recall and the position of the helices, and if both of those are 100%, so if we don't overpredict any, if we captured all of them, if we found all of them, then the whole protein is correctly predicted, and we just count in QOK how many proteins are correctly predicted. To do that, we need to have some kind of notion of when is a helix correctly predicted. So we could say, okay, if every residue in the helix is exactly where we observed it in the database, but that's too strict because the databases are not that accurate, the experiments are not even that accurate. So what we do, same as in the evaluation paper, we want that the helices overlap at least by 50%. And that the ends of the helices do not differ by more than 5 residues. So if that's our observed here, then this helix could be shifted up to here, or even in both directions. As long as it's not more than 5 residues, it's fine. And as long as it overlaps at least 50%, which is comparably much stricter than what was used traditionally. So in older papers, they often had like 3 residue overlap, and that's it, which then would give you 2 helices like this, would still be considered correct, but that's an issue, because if there's already the next helix, then you're almost missing a helix. So that's way too permitting. This, we believe, is relatively strict, but biologically makes the most sense. Of course, then the scores will all be lower than what you're used to, but you've already seen the evaluation, so you know what kind of the baseline is. Yeah, so that's what we used here as well. And then QTop, so since we now have topology, it's the same thing. We look, is the whole protein correctly predicted, but is the topology also correct? So essentially, is the N-terminus correctly located? Because we already know the QOK has to be 1, and then if all of the helices are correct, then only the N-terminus has to be correct, and then the topology is also correct. So that's QTop, which then again is even stricter, if you will, because it requires one more thing to be correct. Okay, and that's the result. I'm going to drink something. Yeah, the methods we chose here, MEMSAT, SVM, and Polyphobius. you remember from last time maybe, because they are more or less what we determined are currently the best methods there are for this task. And then additionally MEMSET 3 we used because that happens to be relatively good at topology prediction, although it's already relatively old. Okay. And as you can see, the absolute score for TMSEG, if we look at the helices, is the whole protein correctly predicted, is actually the highest. So that's nice to see. However, since we have such a small data set, the error bars are so large that we can't really claim that we are significantly better than the other methods. We're not worse either, it's looking pretty good, but yeah, that's about all we can say if we are honest to ourselves. If you look at topology, we can actually see that not only is it again the highest score, it also is significantly better than Polyphobius at least. And it's, yeah, so generally also a good deal higher than the MEMSET group of predictors. So that's good. Then next, the problem is that you do not apply such a method only to membrane proteins. So you don't usually, well, you may know what your protein is, but if you, for example, predict the whole human proteome, you don't know which proteins are membrane and which aren't. So you also have to recognize what is a membrane protein in the first place and what isn't. And that we're measuring simply by the false positive rate and sensitivity. So how many do we overpredict? How many of the true transmembrane proteins do we find? And that we now also compare to a simple baseline predictor just to have an idea of what the background is. This baseline predictor is the most stupid predictor you could possibly imagine. It's actually surprisingly good, but I did my best to make it stupid. So all it's doing is it's using a sliding window, it's using the hydrophobicity scale, Eisenberg I believe, but it doesn't really matter. And as soon as in that window the hydrophobicity goes over a certain value, 4.0, which is empirically determined, it predicts that part as a transmembrane helix, and then it moves on to the other part of the sequence. And that works actually extremely well, which just shows us again that hydrophobicity is an extremely important feature. So that's what we, you know, that's the most simple thing I could imagine, and that's what we use as our baseline. And then this is the result. As you can see, sensitivity, pretty much all methods are good at finding the transmembrane proteins, also the baseline predictor. I mean, it just looks if anything is hydrophobic, as if even one helix is found, then okay, this is a transmembrane protein. However, if we look at the false positive rate, we can see that it also finds other hydrophobic stuff, which it then predicts as a transmembrane helix, so it's not very good at excluding those. And neither are actually MEMSET SVM and MEMSET 3. Only Polyphobius and TMSEG are doing relatively well in determining which proteins are soluble and which ones apparently are not. And then if we also look at the topology, we can again see that TMSEG is the highest performer, and not only that, now Polyphobius actually does relatively bad. So if we were to aggregate the first 3 columns, we could claim that actually TMSEG is giving us the best trade-off among all of these. It doesn't overpredict very much, it finds all of them, and the topology tends to be correct. Yeah, and just to give again an idea of like what our background here is, if we were to apply this to the human proteome, then we can see that using TMSEG would give us the fewest misclassifications, while using our baseline predictor, for example, would give us an order of magnitude more misclassifications in both classes. So as well transmembrane proteins as well as soluble proteins predicted in the wrong class. Okay, then evaluation done. We want to write the paper. This, I'm sure Burkhard has mentioned before as well. 2.5 years have passed since we started working on this, and we want more data always, especially for transmembrane proteins. So we can check what happened in the 2.5 years since we froze our data set. Did anything new get published? And he did this, and 12 new structures came out, 12 new unique structures, of course, that are not similar to the others, and not similar to themselves either, which is all right for 2.5 years, but unfortunately still a very small data set. Looking at these, we can see that TMSEG unfortunately does not recognize all of them as transmembrane proteins, so only 10 are predicted as transmembrane proteins. However, at least among those 10, it actually gets all of the helices correct. So all of the proteins are correctly predicted, which was very nice to see. And also the topology is almost always correct, which is nice. Okay, and then more things. So the method has, we believe, value over just as a method in itself, because the steps 3 and 4 are actually not something that is specific only to TMSEG. So to repeat again, the third step only looks at the done transmembrane helix prediction and tries to improve it by seeing, can we split helices, can we adjust endpoints, all of those things. That we could apply to different methods as well, to the output of different methods as well. And the same for the fourth step, it just predicts the topology. So some methods don't even predict topology, or even methods that do predict topology, we could see if we apply our machine learner to the output of those methods, what happens. And we can see here that for the QOK, performance is improved. So this is the difference in QOK now. Which means if, so this means that we can improve QOK by 10% in MEMSAT3 just by using the third and fourth filter step at the end of the prediction. MEMSAT3 also being the oldest method, maybe that's what we expected, but still it's very nice to see that just using it in a completely different method. I mean, you could argue, for example, that the third step, which improves transmembrane helices, has learned the idiosyncrasies or the errors that the first two steps do, and then improves those in particular. But we can see that also for completely different methods, this actually does improve performance. And even more so, topology prediction is doing really well, improving all other methods. MEMSAT-SVM as the fewest, but all of them are improved in some way. So not only do we have a new method, we actually also have a part of the method that is of value to other methods if you were to use those. Okay, yeah? Question or just, okay, yeah, do have.
So is the performance of the other methods now better than yours when you increase it by like 10 QOK?
That's a very good point. I'm actually not sure how you calculate this, but it should, for the topology, it should at least be the same now, yeah. So yeah, I mean, it's a fair enough point. Which is particular, so I do not have a good explanation why in particular for topology it works so well. Their data is not that scarce, but apparently the filter that we built for this is doing particularly well. Oops, bless you. And the only thing that we can't do, which would be a potential extension, is reentrant helices. So again, not sure if this has been mentioned before. This is a transmembrane protein, it's aquaporin. This is a normal transmembrane helix. It actually crosses the membrane. It goes from blue to red. This is a reentrant helix. It does not do that. It starts here, then it goes to like half of the membrane, and then it goes back here again. So it ends in blue again. So these helices do not cross the membrane. They to some degree go into the membrane, but they never come out at the other side. They actually turn around inside in some way. And that's an issue, of course, because we are at all times for this predictor assuming that every helix crosses the membrane. That's an issue A, because these can be much shorter. So here 7 residues that we've before kicked out, there are reentrant helices. I mean 7 is really borderline because you also have to be sure where the border of the membrane is. But you could argue that there are reentrant helices that are around 7 helices long. And there's also longer ones. So that's one issue. We would not capture these, even if we would not kick them out. Say it's 10 residues long. It's unlikely that our predictor, which has only seen long helices, would predict something like this correctly. And the other issue is for topology, of course, because this does not change topology. It starts on the side and it ends on the same side. So that's really important to also get an idea of the function of the protein, that we would find these types of helices. The problem is we already have a very small data set, and then the reentrants are again not that common among transmembrane proteins, so this is a really small data set. I would have to ask him about the exact number. I believe it's like 15 helices or so in the complete data set that are reentrant. And training a predictor on 15 input, that's really, we did not want to go there, but hopefully we will soon get more data, and then that would be an extension of the method. For example, looking at, as I said, helices that kind of look strange in their length maybe, or that have signals that are not as the others, and then we could check, okay, if we now change topology, does that actually improve the score? Maybe this then is a reentrant helix. But for that, we just need more data. Okay. And finally, yeah, so that's just a general point as well. Your method is of no value if no one can use it. So we have to make sure that it's available. Ideally, something like that. So we have a Debian repository at RostLab, which means you just have to add this as a source, and then you can use apt-get install TMSEG, and it works, which is great. Or you can get the source code and compile it yourself from GitHub. And then finally, of course, it's also integrated into our Predict Protein web server. So that's part of the output from Predict Protein down here. Yeah, you just get your sequence up here, then the annotation of transmembrane helices, and here it tells you that the source of that is TMSEG. So we used to have our old method in there, but now it's been superseded by the new TMSEG. And with that, I'm done. Thank you. Yeah.